Hello, everyone. Welcome. My name is Janet Lee, and for the last 5 years, I had the privilege of serving as Executive Director of the Kabuki Syndrome Foundation. I am also the proud mom of my 16-year-old daughter, Emily, who has Kabuki syndrome. Thank you so much for being here. I was just looking at the chat, and it's just so amazing to see so many families representing globally. We're so glad to come together as a community united by one goal: to reshape the future for individuals living with Kabuki syndrome. Just a few quick housekeeping notes before we get started. We really want this to be an engaging opportunity for all of you and encourage you to use the chat to connect, share what excites you. Also, don't forget to add your questions into the Q&A. Please feel free to upvote ones that you want answered. Also, if you're joining on a mobile device, we recommend that you turn your phone sideways for the best view. And finally, just as a reminder, this webinar is being recorded. So today's webinar, we are so excited to highlight how communities like ours are transforming the rare disease landscape, not only by raising funds and enabling research, but by stepping into a central role of driving treatments forward. We're going to share how far we've come together this year, the milestones that reflect our collective effort, and the path ahead. You go to the next slide, please. So this dashboard highlights the key milestones we've achieved to date. Each one is a reflection of the progress we've made as a community and the foundation we're building for what comes next. It truly takes a dedicated group of affected families to join together to raise awareness and funding for a rare disease. Thank you for making this progress possible. It's your urgency and support that have been so essential in driving progress. I'm so excited to share that KSF has invested $2.8 million in 16 research grants, supporting 25 investigators, all working to advance Kabuki syndrome research. These initiatives address critical steps across drug discovery and development, everything from deepening our understanding of Kabuki syndrome to moving promising drug candidates closer to clinical trials. I just want to note that this $2.8 million does not reflect several vital initiatives that are being led directly by our staff that don't come in the form of grants, but rather through the in-house expertise of our research team. We're driving efforts like research recruitment, patient data collection, and improvements in diagnoses and clinical care. These are all activities that not only advance our research roadmap, but they also directly affect patient families, of which we nearly have about 1,300 families in our Kabuki count registry, representing 66 countries. Having access to diagnosed families has enabled us to recruit over 500 patients to participate in critical research initiatives. Similarly, our network of clinical experts and researchers has grown to 212, with investigators in 15 countries actively involved in KSF-funded research. Your continued support has made all of this possible. Next slide, please. So this year, KSF prioritized 3 critical areas. First, accelerating potential therapies from the lab to the patient. We've invested more than $500,000 in promising research, fueling drug candidates and pushing them towards clinical trials. Second, really focusing on building clinical trial readiness through the success of KSOC Phase 1 recruitment. We're so excited to report that more than 150 patients have now been enrolled across 6 leading institutions, creating the data and tools needed to measure treatment outcomes. And third, improving diagnoses and clinical care. These are 2 priorities that guide research toward what matters most to families. Our research team is going to be spending some time throughout this webinar diving deeper into each of these initiatives, again, reinforcing KSF's role, not just in enabling research, but really driving treatments forward with urgency and purpose. Next slide, please. So in just a few short years, KSF has achieved remarkable progress, raising nearly $5.5 million. And it's all because of the shared commitment of our donors, our families, the staff, and board. This graph illustrates the power of resourcing. Every time KSF has added capacity, we've been able to expand our outreach, raise more funds, and advance our research and community programs. Our board of directors provides oversight and guidance, contributes professional expertise to advance our mission, and plays a vital role in supporting fundraising efforts. The addition of dedicated staff over the last few years has been absolutely crucial, not only to manage the day-to-day operations of the foundation, but also to build and execute a strategic research roadmap. If you take a look at this, you can see that from 2021 to 22, adding an executive director and a director of communications helped grow our revenue from $9,000 to nearly $1 million. Between 2023 and 2025, expanding our board and adding a research director, chief science officer, and associate director of research and community engagement enabled us to define a clear research strategy, raise over $4.5 million, and execute a robust plan to move treatments forward. Additionally, our Kabuki Count Contact Registry has continued to grow year after year, proof of how far our outreach has come and how strong our community has become. I'm thrilled to share some recent changes that will fuel the next phase of our growth. Because fundraising is so critical to our mission, we have added a development director, Makai Gunn, a fellow Kabuki mom and a fundraising expert who you'll be meeting shortly. We're so thrilled to have her join us. In addition, Patrick Dunbar, our co-founder and longtime board member, will be stepping into the role of executive director, and I will be transitioning to the board where I'll continue to provide oversight and strategic direction. It has been an incredible 5 years. Together, we have built a strong foundation, and I really couldn't be more excited to step into this next chapter with Patrick leading the way. At this time, it's my honor to introduce our new executive director, Patrick Dunbar.
Thanks, Janet. First, I want to offer my heartfelt thank you for all that you have done as executive director over the last 5 years. You have put tireless effort into all that you have done, and without that, we would not be here today. So again, I am profoundly grateful, and I do look forward to working with you in your new role on the board and hopefully for many years to come. When we founded the Kabuki Syndrome Foundation in 2018, we were simply trying to understand what research was being conducted, identify who was doing it, and get a seat at the table by funding some small research projects. But it quickly became clear that with high development costs, a small patient population, and limited research funding, finding treatments for a rare disease is uniquely challenging. We have witnessed pharma companies reach out to our Kabuki syndrome community for support, only to deprioritize and shelve potential therapies when faced with these challenges. Fueled by our sense of urgency to find treatments, patient groups like ours must play a crucial role by building awareness, raising funds, driving data collection, and fostering collaboration between researchers and clinicians. KSF has been doing this with your support by raising funds to build KSOC and other foundational capabilities, launching and growing Kabuki Count, as we've just discussed, surveying caregivers and those with Kabuki syndrome to identify treatment priorities, and expanding our network of world-class research partners, including both academic and commercial institutions. And because of this work, the KSF team has been able to drive research forward. Most or all of you are probably familiar with this chart showing the potential treatments we have in our pipeline. Over time, KSF has continued to add programs to uncover potential drugs, small molecules, and nutraceuticals that can be repurposed, as well as exploring new therapeutic technologies like gene therapy or ASOs. During 2025, we have been working directly with our research partners to accelerate 7 of the potential treatments you see here. Notably, we are working with world-class partners around the world in Spain, Chile, Greece, Italy, France, the UK, Canada, and the US. We will continue to work with institutions wherever they may be that are best in class and that give us the greatest opportunity to accelerate new and identified therapies toward clinical trials. As we often say at KSF, we are pursuing this diverse and growing portfolio of potential treatments because we want as many shots on goal as possible, knowing that not all of them will make it successfully to or through clinical trials. Next slide. Just to orient everybody to the agenda for today, our research team will start off by sharing our research milestones that we have achieved and some treatment updates. Then Charlene Son-Rigby, our keynote speaker, will speak about the importance of patient advocacy groups in transforming the rare disease landscape, followed by a Q&A session moderated by KSF Chief Scientific Officer Bruce Bloom. And then Makai Gunn, our new Director of Development, will share her story and discuss how we can raise funds to drive research forward. With that, I will turn it over to Amanda Gamboa.
Thank you, Pat. I am Amanda, our Director of Communications and a Kabuki syndrome parent. I'm thrilled to share that starting October 1st, 2025, Kabuki syndrome will officially be recognized in the U.S. medical records with its own ICD-10 code, Q89.81. This can unlock data for everyone, but we need your support. So ICD-10-CM is, on the next slide, the United States version of the International Classification of Diseases. This system is used to label and track information about all conditions and symptoms at medical appointments, and codes are also used for billing and insurance claims, so you may have seen it before on your explanation of benefits. While ICD-10-CM is specific to the US, the powerful data that we can collect here can also influence international healthcare. Because every single symptom has a diagnosis code, doctors, researchers, insurers, and policymakers can use this data. But until now, providers were using a catch-all code for Kabuki syndrome, Q89.8, which included Williams syndrome, Stickler syndrome, and a dozen more. This prevented anyone from being able to extract Kabuki syndrome-specific data from medical records. But now, starting tomorrow, with Q89.81 for Kabuki syndrome, we've enabled the doctors, researchers, and policymakers to improve their care coordination and your access to clinical guidelines, data insights like how many people have Kabuki syndrome, also known as our prevalence, the range and severity of symptoms, and which treatments, testing, and services are being used by our families. Our unique ICD-10-CM code can also streamline insurance billing and approvals and demonstrate to policymakers that the cost of care is high, unlocking more funding and motivation to accelerate research for new treatments, because more people identified can mean more resources granted, but only if the code is used. A recent study found that the unique code was missing in over 75% of the charts of one of our disease cousins. Correct use varied widely by institution and even within departments of the same institution. We need Q89.81 to show up every time, and we're asking you to advocate for its use so our medical teams can adopt it sooner and accelerate its impact. Together, we can be proactive and make our code count. So tomorrow, the code goes live, and we're asking you to tell all of your providers at your appointments for the next 2 years. You can print these wallet-sized cards, cut them out, give them to every provider, from your pediatrician to your therapist to your urgent care emergency doctors. If the provider that you hand this card to isn't sure what to do, you can just ask them, please place this card in my chart or add a note for the coding team. You can also follow up after your appointment. Did you see the code in your explanation of benefits? Can you find it in your electronic chart? If it's missing, please send your provider a note, and you can always reach out to us with questions or for support. We're going to place the link to the cards in just a minute, and we'll answer more questions during the Q&A. But really quickly, I want to thank you in advance and also those who support our work to get this code. We led a 2-year campaign to secure this code because we know accurate recognition is essential for improved care and research. Our team worked tirelessly to educate decision makers at the Centers for Disease Control, build consensus in the medical community, and ensure that the voices of families and clinicians were heard. We especially want to thank the caregivers and adults who provided letters of support. Dr. Margaret Adam from Seattle Children's, who presented at the Centers for Disease Control, our advisory boards, and professional organizations like the American Academy of Pediatrics and Rare Disease Caucuses in multiple states. Together, we made this possible. Moving forward, every use of the code builds visibility, strengthens insurance coverage, and accelerates the development of new therapies. Kristen, thank you. I think she's going to add the link to the wallet cards in the chat now. And thanks again for your help, everybody, all of our volunteers in the chat make this possible. All right, you guys can reach out anytime with questions. I'll also be back for the Q&A after our next two speakers. And at this time, I'd like to welcome Jessica Weatherstone.
Hi, my name is Jessica Weatherstone, and I am the Associate Director of Research and Community Engagement here at KSF. I'm also a parent to a child with Kabuki syndrome. My daughter is named Emma, and she's 11 years old. And I am so excited to speak with you today about the results of our 2025 community survey. KSF has a long history of supporting patient-centric research. And what that means is that we want to support the development of treatments that our community not only wants, but needs, because there's no point in developing treatments no one wants to take, right? So one way that we can do this is through crowdsourcing surveys. These surveys allow us to identify which symptoms have the greatest impact on the people living with Kabuki syndrome. They also help us to establish priority research areas and then the needs that we identify in the community. We can use this evidence to garner support from our industry partners. Once we have the data, KSF can use this information to shape our roadmap and guide strategic decisions to make sure that we are serving the needs of our community. Our most recent survey was titled Community Survey to Assess Symptom Impact and Treatment Preferences in Kabuki Syndrome. And I know that's quite a mouthful, but the overarching goal of this survey was to understand which symptoms are most affect our community. So our survey was developed with the support of Costello Medical, and we also involved industry advisors, clinicians, and community members as we developed the questions. We distributed the questionnaire via an electronic form that people could fill out on their phone, their computer, or their tablet. And we collected responses from both caregivers and adults with Kabuki syndrome. We ended up with 247 responses. 231 of those were from caregivers and 16 were from adults with Kabuki syndrome. The survey assessed symptoms in 12 different body systems. And I know looking at this slide is a lot, but Kabuki syndrome is a lot. And I know most of you have seen specialists in many, if not all, of these categories. So we really wanted to catch the breadth of symptoms that people with Kabuki syndrome are experiencing. But we also wanted to catch the depth. So we couldn't ask every single symptom that could possibly affect a person with Kabuki syndrome. So we focused on 5 key areas and asked some more in-depth specific symptoms in those areas. And I want to draw your attention to the neurosymptoms. Remember, these questions were developed by experts and community members, so we knew that neurosymptoms were going to be very important. And for neuro, we really went into depth asking about this whole list of symptoms here, including intellectual disability, low muscle tone, speech, social situations, and on and on. And we'll go into these detailed symptoms in a moment. But first, I want to show you the data from these broader body systems. So the first question we asked was which symptom the person with Kabuki syndrome was currently struggling with. And we wanted to know what they were currently impacted by because we wanted this snapshot in time of what our community was struggling with at any given point. So when we look at the data, we see that shockingly, yet not surprisingly, 100% of people selected at least one of those neurosymptoms that I listed before. And then 2/3 selected endocrine, gastro, vision, and about half selected hearing, immune, and dermatological symptoms. Less than half selected cardiorespiratory, urinary, blood, and liver problems. So as you can see, there's kind of a range, and with some symptoms impacting more people than others. This data allows us to focus on those body symptoms that are affecting the most people. So let's take a look more in depth at these neurological symptoms that are affecting so many people in our community. While experiencing neuro symptoms was universal, there was variability in the occurrence of the specific symptoms that we pulled in our survey. Low muscle tone and intellectual disability, speech, social difficulties, and sensory processing impacted the vast majority of people. Anxiety, behavior issues, and sleep difficulties impacted the majority, while depression, migraines, and seizure were impacted the minority. This level of detail is helpful as we are planning our research priorities. The next question that we asked was, out of all of those symptoms, if you could just pick one to develop a treatment, which would you choose? And out of all of the symptoms, the majority of people selected learning and intellectual difficulties. So I think you will see later on in the presentation that this is something we have chosen to focus on as we guide the preclinical testing of several therapeutics. Low muscle tone and coordination, speech and communication, and anxiety were all selected fairly frequently, and we are using these to inform our development of our outcome measures. The rest of the symptoms were selected fairly infrequently, and you can note that less than 1% selected those symptoms that you can see in the box. So while these results might not be surprising to most of us in the community, KSF is currently using the data to guide strategic decisions. Biostatistician is performing a sub-analysis by sex, age, and genotype so we can see if there's any differences in those groups, and we plan to publish the results in a peer-reviewed journal. I will say that this data is being used right now to help guide decisions. Just the other day, I was in a meeting and the researchers were wondering if the speech differences they see in Kabuki syndrome actually impacted daily life. And I was able to pull this up and say, yep, 90% of our community members say it's impacting their daily life. So I know that filling out these forms is tedious and time consuming, but your participation is important and this data is powerful. We thank everyone who has participated because we can't do this without you. With that, we'd like to move on to our research update, which will be presented by our Director of Research, Dr. Clara Tang. Clara?
Thank you, Jessica. So, hi everyone, I am Clara Tang and I'm the Director of Research at KSF. And I'm excited to be giving you a research update today. And I will start by going over some highlights from our research program this year and then discuss some of the research grants that we have funded to accelerate specific treatments in our pipeline. So I'm going to focus on 4 of our research programs: Clinical Management Guidelines, KSOC, which is our Biomarker and Outcome Measure Consortium, our Research Toolkit, and Kabuki Count, which is our contact registry. To start us off, let's dive into the Clinical Management Guidelines. So one of our biggest research projects this year is the development of Clinical Management Guidelines for Kabuki syndrome. These will give doctors everywhere a clear roadmap for how to care for people with Kabuki syndrome, even if they aren't specialists. So this work started more than 2 years ago and has grown into a global effort with experts and families working side by side. Last year, we brought together clinicians from around the world to review all the published medical research on Kabuki syndrome and draft recommendations. And we made sure that the community's voice was included at every step. Then this May, KSF hosted a consensus meeting where clinicians and 12 community members came together to combine medical expertise with lived experience. The guidelines will cover all major areas of care and include over 100 recommendations. We're now finalizing the manuscript with a goal of submitting it for publication next year. This will then be followed by a patient-friendly version that families can use. Next, I'd like to give you an update on how we're developing biomarkers and outcome measures through KSOC, our Kabuki Syndrome Biomarker and Outcome Measure Consortium. So, as Pat mentioned, we currently have 9 therapies in our pipeline. But for any of them to actually reach patients, we need to show that they're effective. And right now, there are no validated Kabuki syndrome specific biomarkers or outcome measures. And these tools are essential. They are what allow us to measure whether a treatment is effective, attract industry investment, unlock new funding, and ultimately move more therapies towards patients. And thanks to your support, in 2023, we raised $1.35 million to launch KSOC, a consortium dedicated to validating Kabuki syndrome specific biomarkers and outcome measures. The team is currently focused on 3 types of biomarkers: the DNA methylation signature, metabolomic markers, and event-related potentials measured with EEG. Many of you in the audience may have already taken part in KSOC by donating samples and sharing your data, And all of these measures will be compared with neurocognitive testing results to identify patterns that can serve as reliable, meaningful ways to measure treatment efficacy. And we are proud to announce that with your help, we have reached our goal of recruiting 150 families to participate in KSOC across 4 sites in the United States. The last few families are completing their appointments in October, and we expect to begin data analysis soon after. We are deeply grateful to everyone who has taken part in this project, and we could not have accomplished this so quickly and seamlessly without you, our dedicated and supportive community. Next, I would like to share more about our research toolkit we have been developing. This resource is designed to accelerate both current and future therapeutics in our pipeline by providing scientists with access to valuable tools to advance the study of Kabuki syndrome. So our virtual toolkit is housed on our website and marketed directly to researchers. You can explore some of these tools through the link that Kristen will put in the chat, or by scanning the QR code on your screen. And I'd like to highlight 4 of the tools we have been developing. So first, our cell line bank. We are establishing several Kabuki syndrome cell lines that will be stored and shared widely with researchers at little or no cost. These cells are derived directly from patients with Kabuki syndrome, making this project particularly exciting. And once available, labs can use them to study the molecular biology of Kabuki syndrome and test potential new therapeutics in a human-derived system. And later in the webinar, we will share more details about our fundraising efforts for the next step in this project. Next, our mouse models. We're working on establishing 2 Kabuki syndrome mouse lines that will be available for experiments and tissue collection. One line is already established at a contract research organization, CRO in the Netherlands, and a second colony is being set up at a lab in Chile. And I'll share how we're using them to accelerate the therapies in our pipeline a bit later. And we are also then curating and publishing protocols specific to Kabuki syndrome and our genes of interest, KMT2D and KDM6A. So for example, we worked with a lab to test commercially available antibodies for KMT2D, determine the best one, and write up a detailed protocol for its use. This allows other labs to skip the trial and error process and immediately apply the best methods. And projects like this maximize efficiency, reduce redundancy, and accelerate progress towards treatments. Finally, many of you are already familiar with Kabuki Count, our contact registry. This is the largest contact registry for Kabuki syndrome worldwide. It not only tracks how many people have Kabuki syndrome, but also provides a way to reach them. This has enabled us to recruit for our own clinical studies, such as KSOC, and to support external researchers. In fact, it has allowed us to proactively approach experts in other fields and encourage them to study Kabuki syndrome by making participant recruitment seamless. For example, we recently recruited for a visual spatial study led by a vision specialist who had not previously studied Kabuki syndrome. But he took on Kabuki syndrome as one of his new areas of expertise because we were able to provide him with immediate access to interested participants. And as of last quarter, we had just under 1,300 people in the registry across 66 countries. And if you have not yet joined Kabuki Count, we invite you to do so now. Kristen will drop the link in the chat, or you can scan the QR code on your screen. We also need your help spreading the word. When you meet newly diagnosed families, please encourage them to join. Growing this registry is something that we can all do together to unite our community under one common goal. Those were just a snapshot of some of the research projects happening at KSF. And now I'll shift to give you an update on our therapeutic development pipeline, and starting with an overview of the therapeutic development process. So to get an effective treatments to patients, we have to move through several steps. We need to learn more about Kabuki syndrome, like through our Discovery Grant program, build tools such as the biomarkers and outcome measures being developed in KSOC, test therapies and models like the cell lines and mouse models I described in our research toolkit, then test the most promising therapeutics in people through clinical trials. We're not quite at this stage yet, but we are laying the groundwork with projects like Kabuki Count so that when a therapy is ready, we can move quickly to recruit participants. Then finally, if the data show safety and efficacy, results are then submitted to regulators like the FDA, leading to hopefully approval and ultimately access to patients. So the process I just described supports all the therapies in our pipeline, which are the treatments we hope to move into clinical trials. Right now, we have 9 therapies in various stages of preclinical development. So on the left, you can see the list of potential treatments, and along the top, their stage in the development process. Green arrows represent progress made independently of KSF, while yellow arrows highlight progress funded by KSF. And as you can see, all of our therapies are still in the preclinical stage, but we are steadily moving forward. And today I will focus on the treatments marked with the yellow arrows to give you an update on the work that KSF has supported. So I'd like to start with our leading candidate, fepidemstat. So fepidemstat is a drug developed by the biotech company Horizon that targets a pathway that could potentially benefit people with Kabuki syndrome. To determine if this drug is effective and to help to risk future clinical trials in humans, KSF has funded a 300,000 preclinical study in mice. So the first step was establishing a Kabuki syndrome mouse colony at a contract research organization running the study. This took some time, but I'm happy to share that the colony is now established and breeding. And this was an important milestone because this colony becomes a resource that we can use for future research studies. As part of setting up this line, we needed to characterize the mice and identify a behavioral task where Kabuki mice show a clear deficit. Based on the crowdsourcing survey we shared, as Jessica shared earlier, we focused on task testing, learning, and memory. And after months of trialing different options, we think we've found the right task and are now working to verify those results. Since fepidemstat is given orally, we also wanted to confirm that the drug actually reaches both the blood and the brain, and our results suggest that it does. And from here, we will look at biochemical markers like methylation signatures, RNA sequencing, and indicators of new brain cell growth to see if there are any cellular level changes. Then finally, we will assess whether the drug improves performance on the behavioral task. And with additional funding, the next step is to test fepidemstat in human cell lines at Dr. Rosanna Wexford's lab at the University of Toronto. All these results will guide a go, no-go decision on whether to begin clinical trial planning in humans. And I hope to have the results of this study next year. Next, I would like to move on to another promising therapeutic that is in the therapeutic development stage, the modified Atkins diet and BHB or beta-hydroxybutyrate. As many of you know, several pilot studies have been conducted with people with Kabuki syndrome using the modified Atkins diet, which is a diet that is low in carbohydrates and high in fats and proteins. The evidence from these studies is promising, and the hypothetical mechanism of action is that these diets increase the levels of beta-hydroxybutyrate, BHB, in the body, and anecdotal evidence has shown that higher levels of BHB could influence cognitive improvement. Because the lifestyle changes required for these diets can be challenging, we wanted to investigate whether raising BHB levels through exogenous BHB supplements could be sufficient to produce therapeutic effects. The challenge with these supplements is that BHB levels spike briefly after each dose and then decline until the next dose. So whereas the modified Atkins diet produces more sustained BHB levels. So we need to determine whether these transient BHB spikes can have the same therapeutic effects as sustained levels. And so to test this, we started with mice. We're establishing a mouse model at a lab at Chile, which will be our second model ready for use in this experiment and future studies. Next, we're working on the dosing schedule, so determining both the dose and frequency required to raise BHB levels in the blood and brain. Once that is established, we will test whether these doses alter neuronal function or improve performance on behavioral tasks. And this work is aiming to start by the end of the year. And if we do identify a therapeutic effect of exogenous BHB, we will move forward with planning for a clinical trial as early as 2026. And next, I would like to discuss 3 of the candidates in our pipeline that all use a similar method, drug repurposing. So drug repurposing means taking drugs or therapeutics that are already on the market and using them for a new purpose. The advantage is that if these drugs are already proven safe, the path to patients can be much faster and less expensive. Our goal is to identify and develop existing FDA-approved drugs or nutraceuticals that have the potential to treat Kabuki syndrome. And we're pursuing this in 3 ways. So let's first talk about the project at Boston Children's Hospital, BCH. So we invested $110,000 in this effort to test candidate compounds that can boost either KMT2D or KDM6A in Kabuki syndrome human cell lines, and then move into our mouse model. The team has already found some promising candidates, and right now they're finishing the cell line experiments and beginning testing in mice. We also invested $100,000 in a collaborative project between Purposeful and Gen. This project uses AI to predict and prioritize candidate drugs that could improve Kabuki syndrome symptoms. And finally, we funded $66,500 in a project with Unravel Biosciences, which is focused on Kabuki syndrome type 2 caused by mutations in KDM6A. And the goal with this project is to identify potential repurposed drugs by analyzing patient samples. The collection of the samples is now complete, and so thank you to everyone who has donated your nasal samples, and we expect the report on candidate drugs within the next few weeks. Our next steps will be to continue testing the most promising candidates, especially those that identify across 3 repurposing approaches. By the end of these projects, we hope to add or progress several rows in our therapeutic pipeline. So far, the therapies we've talked about are drugs, nutraceuticals, or dietary changes. And now I'll briefly touch on our most recent discovery grant, which was awarded to David Genevieve at the University of Montpellier for his work on DCAS9, which is a gene targeting tool. So DCAS9 is a tool that can be used to increase gene expression. In Kabuki syndrome type 1, there is not enough functional KMT2D, so we're exploring the use of the DCAS9 system to restore KMT2D to functional levels. So first, the team will design a KMT2D specific DCAS9 system. They have evaluated the gene and believe they have developed a system that can effectively target our gene of interest. Next, they will test the system in zebrafish to assess its safety and to confirm that it successfully increased KMT2D expression. Ultimately, we want to know whether this tool can improve learning and memory. So the next step will be testing whether KMT2D upregulation enhances zebrafish performance on a learning and memory behavioral task. So it has been a very exciting year with new programs added and existing programs moving forward. I know we went through the pipeline very quickly, so I'm happy to answer any questions during the Q&A. And at this time, I would like to introduce Dr. Bruce Bloom, our Chief Scientific Officer, who will be moderating the Q&A.
Thank you, Clara. Nice job for everybody who's presented so far. I know we've provided a lot of information in a short period of time, and glad to take some of your questions. So just to reiterate, I'm Bruce Bloom. I'm the Chief Science Officer here at Kabuki Syndrome Foundation. And my job is to assist the rest of the team in making sure that we're accessing the research and clinical network around the world, and to provide my 20 years of plus of expertise in therapy development in the rare disease space. So first question is for Clara. The question is about CRISPR technology. How do we feel about CRISPR technology?
Yes, so CRISPR is a gene editing technique, and we're very excited about the potential of gene-based technologies like CRISPR and gene editing. But there are some challenges for Kabuki syndrome. Right now, we know that in Kabuki syndrome, every patient's mutation is a bit different. And so delivering a permanent edit safely to the right tissues is currently still some way to go. And that is why KSF currently is investing in a more innovative gene targeting approach like DCAS9, which doesn't change the DNA but can boost the activity of genes like KMT2D or KDM6A. So, as I've mentioned from earlier, for the discovery grant with David Genevieve's lab, the early results in zebrafish seems very exciting, and hopefully we'll have a first readout later this year. And so we'll look forward to sharing more about that soon.
And I'll just add to that, if you've heard about gene therapies in other rare diseases that sound or are similar to Kabuki syndrome, it all depends on which gene it has a variant in it. Some genes are small and some genes are large. And right now, the way we do gene therapy and use CRISPR technology, you have to put the entire new gene into a viral vector, a carrier, kind of like a spaceship that goes into the body, into the right tissue to replace the gene. And the Kabuki, the KMT2D gene, is too big to be put into the virus right now. So we're looking at other ways we might be able to split that gene in two, and then reconnect it back in the body, or other ways. So we continue to look at gene therapies, CRISPR technologies, and as they become more effective and efficient for the kind of mutations we have in Kabuki syndrome, we'll continue to move those forward. Second question for Clara. Curious if a child with dual genetic diagnosis would still be eligible to participate in clinical trials and into potential treatments.
So this will depend on the specific trial. So each clinical trial will set its own inclusion and exclusion criteria. So, and sometimes having another genetic condition could affect eligibility. So that really depends when the clinical trial is being designed, what the people planning the trial thinks about decides what the inclusion and exclusion criteria will be.
Great, thanks. Another question, two questions about the BHB and modified Atkins diet, Clara, that you could probably answer. Does the modified diet work? And do we have a link to information about the modified Atkins diet?
So with the modified Atkins diet, there has been anecdotal evidence that potentially the ketogenic may be effective. We know that Dr. Jacqueline Harris from the Kennedy Krieger Institute have conducted an early phase 1 trial using the modified Atkins diet in adults with Kabuki syndrome. And the results are still very early, and she's looking to get this published. But the overhead top line results are that the diet was safe in people with Kabuki syndrome, and that people with Kabuki syndrome was able to adhere to the diet. But we know that from preclinical studies that the ketogenic diet did show an improvement in the mice. So so I hope that answers some of the questions about whether the modified Atkins diet does work, and that's what currently the evidence show that there is an indication that it could work, and that's why we're continuing the work with the BHB. And with the link for the modified Atkins diet associated with the increase in BHB levels, are you looking for a paper to do so? Well, we can share this probably after the webinar directly with the person who submitted the question. Would that be okay? Or
Yeah, and then once the full publication is published, we'll share it like we always do on social media. You'll see it on our website as well. It's a publication from Dr. Harris at Kennedy Krieger that we're very much looking forward to. And I'm happy to just answer some of the follow-up questions and really quickly just remind everybody, we cannot give medical advice. Unfortunately, that is not our role today. So I just kind of want to broadly address some of the questions about giving your child supplements or starting a new diet. That should always be conducted under the care of a provider. We're going to continue to fund research that shows efficacy and safety in people, but that is in process right now. And again, just trying to hit several. So the improvements that we're seeing in the mice, potentially with BHB, potentially with Atkins, kind of think of them as one pathway or one way to target. The symptoms that we're looking at are neurological improvements. So the mice's memory and ability to complete memorized tasks more quickly. That would be the target right now in the mice, and we're going to explore more how that can translate to humans. And Clara, feel free to science any of that, but I was trying to just give a really quick overview because there's a lot of questions about diets and things like that. And we have what we have right now, and we'll continue to keep you guys updated. We do have time for just one last question.
There's a question, are there any efforts to look at approaches taken to treat other diseases? Are there opportunities to piggyback or combine with other rare disease foundations? Clara, do you want to talk about what we're doing in that area?
Clara's on mute. I'm going to jump in, or Jessica, if you want to touch on this one.
I can answer this. I mean, we are working with some of our sister and brother organizations. There are other diseases that are similar but not the same as Kabuki syndrome. You know, Kabuki syndrome is a chromatinopathy, and there are lots of other chromatinopathies. In fact, the Cornelia Delane Syndrome Foundation is helping to put together an overarching organization around lots of chromatinopathies so that we can all work together. We also, Kabuki syndrome is part of a group called Combined Brain, which is a lot of neurodevelopmental disorders. And so we get advantage of learning from all of our peers. There's about 120 different diseases represented at Combined Brain. So very much so when we're looking at the DCAS9 and the gene therapies, we're working with labs that are addressing other diseases that are similar in scope to Kabuki syndrome. So overall, Jonathan, I think the answer to your question is yes, we're leveraging wherever we can. We share very transparently, and we feel grateful to the rest of the rare disease community that they also share very transparently, which is a great way of transitioning over to our next speaker, Charlene Sohn-Rigby. So let me introduce Charlene, who I've known for over a decade and is one of the really great leaders in the rare disease space. So Charlene's the Chief Executive Officer at Global Genes, the nonprofit organization dedicated to eliminating burdens and challenges of rare disease for patients and families globally. She spent her career building organizations at the intersection of data, technology, and life sciences. She was previously the Chief Business Officer at Fabric Genomics, and she held executive roles at enterprise software and genomics companies, including Oracle and Double Twist. And she started her career as a neuroscience researcher at Roche. When her daughter was diagnosed with a rare disease, Charlene co-founded the STX-BP1 Foundation to help build awareness and accelerate discoveries of new treatments. Her unplanned connection between her personal life and her professional life pushed her towards a search for a cure for her daughter and children like her, and has given her work even deeper meaning. Charlene holds a BA in human biology from Stanford University and an MBA from the Haas School of Business at the University of California. It's my pleasure to introduce Charlene Sohn-Rigby.
Bruce, thank you so much. I'm really delighted to be here and to talk with y'all at your family conference. This is such an important gathering spot and community building opportunity. And so I'm just so pleased to be part of it. What I want to do today is talk about how broadly patient advocates are transforming the landscape of rare disease. I'm appreciative of being able to go after Dr. Tang, where she talked about all of the progress that is happening now in Kabuki syndrome. And what I'm going to talk about today, you're going to see some shared themes. So I always like to start, and if you can go to the next slide, with my why. And so this is my daughter Juno. She just turned 12, and she was diagnosed with STXBP1 disorder in when she was 3. And this was after a several year journey to figure out what was going on with Juno. She presented, as it turned out, after we got her diagnosis, she had an atypical presentation of STXBP1. And she didn't present with seizures initially. And that was the reason why it took us a while to get an answer. I feel very lucky that when we got that answer, it opened up a door. And if you can advance the slide, what that door opened up for us was first a small set of people who we started the STXBP1 Foundation with in 2017. And then that opened us up to a larger community. If you go to the next slide, this is a picture from our first family meeting in 2019 in Philadelphia. And I think that one of the themes that I am going to talk about today is patient advocates and how patients really are able to transform what is happening in a disease. So this is the STXBP1 Foundation. This is how I got my start in rare disease from a patient perspective. But one of the things that I became very passionate about is how can we advance work across many rare diseases. Bruce just talked about how KSF is collaborating with other chromatinopathies, with combined brain, these type of broader partnerships really enable us to accelerate things. And that is really the mission of Global Genes. So if you go forward 2 slides, please. That's really the mission of Global Genes is to, first of all, our vision is to eliminate the burdens of rare disease, and how we are doing that is through equipping and enabling patient advocates to really activate their communities, as well as really advance research. And so why is this important? If you go to the next slide, I start with this because in 2017, when we decided to to change the landscape for STXBP1, this is what it looked like from my perspective. It really looked like Sisyphus having to push this boulder up a hill or a mountain. And what we can do as patient advocates, and also as a broader community, if you go to the next slide, is really change the curve of this of this mountain. And I'm going to give you some examples of groups that are doing that. I want to start with a couple of movies. So if you go to the next slide, some of you may have seen these films, and what advocacy used to be. My examples are going to start in the early 1900s, but what advocacy used to be was, almost superhuman. John Crowley, who Extraordinary Measures was based on, and his work in Pompeii, and Lorenzo's oil. These parents who found a therapy for their child who had adrenoleukodystrophy. These are the things that movies were made of. And if you go to the next slide, I have a set of slides here, and I want you to just focus on the high level things, that the themes that I'm saying, not all of the details here. The details tell you that a lot of stuff happened, and but the story theme is around what the foundations and the patients did. So this first example is for cystic fibrosis, and many of us are very familiar with this disease. It's one of the more common rare diseases. So the first cystic fibrosis cases were reported in 1930. And so, yet it took until the 2018 and really 2019 when 95% of patients had a therapy. And I think about this in terms of the commitment and the drive that these patient advocates had when they started a foundation for cystic fibrosis in 1955. We didn't even know whether there was a gene or what the gene was that was causing this devastating disorder, cystic fibrosis. And yet these families, these parents had this vision that they could change the future for, the face of this disease and, for the patients. And so they, pushed forward. And, for those of you who, some of you may have read Breath from Salt, amazing, book that chronicled this, but, a 90-year journey to develop therapies for cystic fibrosis from the time that the first case reports were published. So going forward, I'm going to give another example. And if you go to the next slide, this is for Angelman syndrome. And Angelman syndrome is a neurodevelopmental disorder, and the first case reports were published in 1965. And what you can see is that in the bold here, the first patient advocacy organization, the Angelman Syndrome Foundation, was formed in 1986. And then the Foundation for Angelman Syndrome Therapeutics was formed in 2008. And when these foundations were started, suddenly progress really started to accelerate. The gene was identified, therapeutic development started, and you can see that in the kind of last 2 or 3 years, multiple gene therapy trials, genetic therapy trials have started, and really significant investment in different therapeutic strategies, including gene editing. And this all happened in a 60-year period, so 30 years faster than cystic fibrosis. Now I'm going to talk about STXBP1. And so if you can go to the next slide, this is the drug development timeline for STXBP1, my daughter's disorder. So the first case reports on STXBP1 were published in 2008. And this was 4 patients who had very, very severe early onset epilepsy. And from that time, several years later, first a foundation in Spain was started, the Asociación Syndrome STXBP1, and then the US Foundation, the STXBP1 Foundation, of which I'm a co-founder, was started in 2017. And we have very much focused on accelerating therapeutic research. So we were, very fortunate that a lot was known about the basic biology. So, STXBP1 is involved in communication between neurons, and it is part of a set of proteins that pushes what's called neurotransmitter out of the cell. And that neurotransmitter is how neurons communicate with each other. And so patients who have an STXBP1 mutation are not producing enough of that neurotransmitter, and that's why they have, you know, a set of significant symptoms, and I'll talk a little bit more about that. But going back to this timeline, once the reason why these patient foundations were started is we had done a lot of research, and people were continuing to do a lot of research on STXBP1 and what the biology was, but no one was actually working on therapies. And so in 2017, the STXBP1 Foundation was started. Our focus was to kickstart therapies, and we banded together with you know, a global set of other foundations and parents to really kickstart you know, the therapy development process. And you know, so we have just started to enter clinical trials, but you know, we have been doing this now for 16 years since the you know, since the first case reports were published. So you can see that there's tremendous acceleration. And you know, I think there's two major you know, major points that I want to make here, which is one, we stand on the shoulders of giants. So STXBP1 is lucky because we have the history of what happened with cystic fibrosis. We have the history of what has been happening with Angelman and with other disorders. And we, you know, draft off of them. We partner heavily with them. And it is because of that that we have been able to accelerate this process significantly. You know, the other thing is that we are in a very, very exciting time. 15 years ago, all of the progress with genetic therapies, with repurposed drugs like what Dr. Tang talked about, was really not in the, you know, the sphere of what was happening regularly with you know, with rare diseases. And so we are in this very, very exciting time where innovation is now turning into real therapies, real clinical trials, and approved drugs. So if you can go to the next slide, this is a picture from our family meeting this year in July. And as I described, you know, our mission, and I really want to focus on the second bullet here, our mission has been to fund and drive research to accelerate the discovery of a cure. So let me talk a little bit about STXBP1. If you can go to the next slide, just so I give this framework. As I had said during that timeline slide, it was identified in 2008 in patients with Ohtahara syndrome. And there is a spectrum of symptoms. Seizures is typically the first symptom that's identified, but patients also have a broad spectrum of issues, including developmental delay and communication issues. And the STXBP1 protein is involved in communication between neurons, really at that synaptic cleft. So if we go to the next slide, I just want to also give you a framework of when my daughter was diagnosed, there were 200 patients who had been diagnosed in the world. And now we know that the estimated incidence rate is 1 in 30,000. So it is still an ultra rare disease, but you know, if you assume even you know, um, a, a lifespan to 50 years, and we have patients who are older than that, we're still talking about a couple 100,000 patients who are walking around. But diagnosis is a challenge for all rare disease communities. And so we have about 1,500 patients who are diagnosed worldwide, yet it is one of the most common diagnoses for patients who are referred for genetic epilepsies, both in the general population as well as in adults. If you can go to the next slide. I'm going to skip through this really quickly. We have, you know, as I described in the timeline side, been, you know, really working to compress time as much as possible. You know, we have urgency. Patients are waiting and a community that is very activated. So if you go to the next slide, I want to talk about the importance of patient voice. So as I said, my daughter has an atypical presentation, and that is why it took longer for her to get a diagnosis. And seizures often present in the first day of life. We have, a large percentage of our patients who are not able to achieve full seizure control yet. If we look at, and this is data from patient families, if we look at the highest priorities from patient families, speech and communication, cognition, movement, both gross motor and fine motor, are significant challenges for patients and patient families. And I would expect that there's some resonance in in this community around this need to really have have a broad set of symptoms be addressed through therapies. This data, and if you go to the next slide, was replicated in our FDA patient-focused drug development meeting in October of 2023, where we had almost 300 participants. And the important thing here was taking the patient voice, and if you go to the next slide, making sure that not only the FDA knew, but that there was published information on this. So this is our disease concept model that summarized this information on the patient experience and is broadly available to researchers. I think that the we talk a lot about patient experience, and these are the ways that this gets communicated to both the FDA so that they are ready for therapies and understanding why the endpoints that a sponsor is interested in are what they are, and so that researchers understand this. So I want to talk to you about since I focused on this idea of kickstarting therapies, I want to give you a sense of how our therapeutic development has grown. So if you go to the next slide, this is what our therapeutic pipeline looked like in 2019. Frankly, it looked pretty bleak. We had a paper from one of our collaborators at Weill Cornell, and they had identified a repurposed drug called 4-phenylbutyrate that was already approved for urea cycle disorders, and they thought that it might work for STXBP1. And we did not have, so this was great, except that we had no money to take this into a clinical trial. So, our community got together, raised money with the STXBP1 Foundation and two other partners. We raised the money for this clinical trial, and that was the first clinical trial for STXBP1. And then if you fast forward, if you go to the next slide, we're going to fast forward to 2023, and this is what our pipeline looked like. We had 10 therapies in pipeline, and you can see that we start to get a diversified pipeline in terms of gene therapies, RNA therapies, and small molecule drugs. And now, if you go to the next slide, this is what our pipeline looks like today, or in Q3. We have 17 therapies in pipeline, and I think that one of the things that I talk with our community a lot is about having as many shots on goal, because we want to make sure that as Dr. Tang talked about, there's a whole process, a whole set of steps that have to happen in order to make sure that the science is good, that the therapies work, and that they're safe. And not all therapies are going to make it through, either because of science or business. And so that's why it's important to, as much as possible, have a diversified pipeline. So, we are still, early in the process, or mid in the process in terms of moving our pipeline through, but we're very, very excited because we are just starting the clinical trial process, and getting, basically starting the process of getting therapies to patients. I want to round things out. If you go to the next slide, talking about how we are engaging. So this is our 3-year plan from 2023 to 2025. This is actually the last year of this particular plan. And we have 4 pillars in our plan. One of them is to propel precision therapies. And, as you saw in my last slide, this is where our focus has been to ensure that we have a broad pipeline and that we are advancing those precision therapies. The second is around preparing for clinical trials. So clinical trial readiness has been a really key aspect for us. I talked in those timeline slides about how all of us are trying to, really accelerate therapy development and in some ways bend time and space. And yet, once we get to clinical trials, we need to know, what are we going to measure? We need to know where the patients are. So ICD-10 codes, congratulations, by the way, on the ICD-10 code. STXBP1 also has just received ours in October, so we are very excited. But our natural history study, there's many, many things in terms of clinical trial preparation that, your foundation is leading and are going to be critical to propelling those therapies. Building biopharma company interest. We have, we invest a lot in terms of business development to get those therapies of interest, and also to continue the development. And then finally, supporting patients today. This is critical in terms of all of the work that we need to do to ensure that we have an activated and also supported community while we're on the path to these therapies. And so I just have one last slide, which is just bringing it back to the community and the criticality of all of the families in this journey. So thank you. And I believe there's a couple minutes for questions.
Thank you, Charlene. We can't turn on everybody's microphones, but you'd hear a big level of applause for that presentation and for all the work that you've done. I guess a really important question is, when you think about broadly and put on your Global Genes hat, what gives you the most hope in the rare disease space for therapy development and care for the individuals that we care about?
Yeah. I think that this is a different time, and Bruce, I think you would agree, since you have been in pharmaceutical development and seen what has been happening over the last 20 years. We are in a very, very different time now than we even were 10 years ago. And I am just so I am palpably excited about what we have started to see in these first genetic clinical trials, so genetic therapy clinical trials for other disorders like Dravet syndrome, like Angelman syndrome that I talked about before. We are really starting to see what I think is the tip of the iceberg of what we will be able to do. So I am extremely excited about that. I also think that one of the things that that is really important, and Wendy Chung said this says this really beautifully in terms of care before cure, that being able to figure out how do you connect these things. And so one example that we've had in the STXBP1 community has been that we have established a natural history study. We now have 6 sites in the US, but the strategy has been that we had no sites. We had one clinic at Children's Hospital of Philadelphia that knew about STXBP1, and we had otherwise no expertise. And so by building those natural history sites and then thinking about them as future clinical trial sites, as well as expert care centers, we are really working to change what that today clinical care dynamic is. And I'm seeing this, kind of across the board in terms of rare.
Great. Well, thank you so much for taking time out of your busy schedule to be with us today. We really appreciated the information that you gave, and all the work that you do both at XTXBP1 and at Global Genes for the entire rare community. And I am going to turn this now over back to Janet to introduce our next speaker. Thanks, Charlene.
Thank you.
Thank you so much, Bruce. And thank you so much, Charlene. We had a few questions coming in the chat, just kind of asking about the connection between XTXPP1 and Kabuki syndrome. And while, they're both neurodevelopmental disorders, I think it's just so important for our community to see case studies like yours to ensure that, I guess if anything, like you mentioned, so many rare disease groups kind of are get advice from these giants in the space. And that's exactly what we've done as a foundation. And to see the level of progress that you've made and to follow hopefully in those footsteps is really encouraging and just really exciting to get your perspective on the recent changes in landscape. So thank you so much for sharing your work with us. So at this point, I'd really love to introduce Makai Gunn. And before doing that, I just want to say, part of everything that you've seen today, so many of our advancements, as we continue to work towards treatments for Kabuki syndrome, fundraising has been absolutely central to our success. It has made it possible for us to fund research grants, to build the vital programs, and to really prepare for future clinical trials, which are in the pipeline. Raising funds, as you all know, is challenging, but it is absolutely essential in order for us to advance our mission, which is why the KSF team strongly feels that dedicated expertise in this area is so incredibly important. I'm just so fortunate to welcome a new leader into our team community. Makai Gunn is a seasoned fundraiser and a Kabuki mom, and we couldn't be more thrilled to have her part of our community and our team. So Makai?
Thank you, Janet. Hello, everyone. I am thrilled to be with you all today. As Janet shared, I am the new Director of Development for KSF. I have over 15 years of nonprofit development experience. I started as an intern at the Leukemia and Lymphoma Society during college, and the rest is history, as they say. Most recently, I was a director at Thread Strategies, a consulting firm focused on helping small nonprofit organizations build sustainable fundraising programs. Thread Strategies was a partner with Chan Zuckerberg's Rare as One initiative over the past 4 years. So I worked with a number of rare disease organizations to help them build their first sustainable development plan and operations. This work of scaling development and fundraising has truly been the focus of my entire career. And similar to Charlene, I also had an unplanned intersection of my personal and professional life when in May of last year, we welcomed our second child, our son, Brody. By day 2 of life, it was apparent that something was significantly impacting Brody. And by week 3, we had our diagnosis of Kabuki syndrome. The very first thing I did was look up to see if there was a nonprofit organization already dedicated to this work. Finding KSF in that NICU room, surrounded by the constant alarms of all of Brody's machines, it truly felt like the outlook of Brody's life changed. Because of KSF, we have been able to drive his care and treatment from the very beginning. Just to share a little bit more about Brody's story, as I know so many of you can relate, we had 111 days inpatient at the start of life between the NICU and a long-term cardiac care unit. And in total, almost 6 months of inpatient time during the first year of his life. Brody has had 6 surgeries, a G-tube, a cardiac surgery, a craniectomy on his skull, bladder, colon, etc. And he has 2 more coming up this year. While day-to-day life is much improved from a year ago, it is still so challenging and so hard, as you all know. What I want to talk about is the hope and our therapeutic development process. We spent a lot of time talking about the drug development pipeline already today and the details of what KSF is driving forward. But what does this actually mean, and how can we simplify this a little bit? I'm not a scientist, I'm a fundraiser. So here's how I think about it. The first step is to learn more about Kabuki syndrome and the ways in which it impacts the body and how the body functions. Next, we build and create the tools And this looks like outcome measures, which we're already doing with KSOC, and in the future, adding a natural study history platform, as Charlene mentioned, they've done. In step 3, we gather the tools and a large set of data points across Kabuki syndrome that we can test in models, such as mice and zebrafish. And this allows us to see how these therapies impact living beings. Then step 4, the step that everyone gets excited about and what you hear a lot about, testing in humans, clinical trials. But to get to step 4, we have to accomplish steps 1 through 3 first. Trust me, I wish just as much as you all do that we could skip right to step 4, but we can't. Finally, in step 5, which is all of our combined goal, we are getting treatments onto the shelves and into the hands of those who need it. As we heard from Clara earlier, any therapeutic development pipeline is complicated and complex and timely. But what's particularly exciting to me as mom to Brody is that we already have a handful of therapies in step 3. This is amazing. KSF has already driven the funding and work behind steps 1 through 3 for a number of therapies in our pipeline. However, we also know that not all therapies will reach step 5. It's just the reality of drug development and therapy development. But KSF is committed to continuing to drive as many therapies as possible to this step. This is why it's critical to have multiple therapies or multiple shots on goal, as Charlene said, as possible in our pipeline. The more resourcing and funding that we can secure, the faster we can drive these therapies into clinical trials and treatments. We have been able to sprint in the past. Charlene talked a little bit about that timeline. We as KSF have been able to sprint in the past few years because of your support, and we are optimistic that we can continue to do this work at the same pace with your ongoing involvement. Rare disease organizations are the engine behind research and treatments. As we've heard from Charlene, this partnership is critical to driving treatments for our population. A lot of what Charlene touched on for her daughter's condition in terms of the pipeline, multiple shots on goal, advancing therapies, and clinical trial readiness mirror exactly the work that KSF is doing right now. Rare disease organizations are often the catalyst that moves a condition from unknown, as you all experience, to treatable, and KSF is doing just that through a variety of channels. What I find phenomenal is that 99.8% of all funding that KSF has raised has come from those who love someone with Kabuki syndrome. Large pharmas rarely invest in the early phases of the rare disease space, and that means that rare disease research wouldn't move forward without the funding that comes from patient-led organizations like KSF. And I'm excited to talk to you right now about one particular way that you all can participate. Starting in 2026, KSF is launching a DIY, do-it-yourself, fundraising program. We will be leveraging Rare Disease Day at the end of February next year, as we know that this is a critical day in all of our calendars. And this is something that you will be able to engage in and execute whenever you want throughout the year. DIY fundraising is a really powerful way to transform our experiences into meaningful action that fuels research and hope. It can give us a powerful sense of agency, proving that our voices and our efforts directly drive progress towards treatments. I know that in so many doctors' offices, I feel a bit helpless, like I can't really control the outcome for Brody. But by fundraising and raising money that directly drives our research efforts, I know that I'm making a difference in his life and his future. KSF will be building an online platform that anyone can use to reach out to their friends and family to donate to KSF. You can simply send out emails and texts, or you can host a little birthday fundraiser. I know a lot of you are already doing this, but this will make it so much easier. You can have a backyard barbecue, all sorts of different ideas and ways that you can get you and your community involved. We will be building out sample emails, texts, toolkits, resources to support you in this effort. And you should also feel free to customize any of these to share your personal story. If you're interested in learning more about how to engage in this new way to fundraise and drive treatments, please feel free to reach out to me if you want to be an early adopter and help us as part of next February's launch. Clara mentioned how many different therapies we have in our pipeline. And one of our next steps is to test some of these therapies on human cell lines. We have set a goal to raise $70,000 that enables us to test certain therapies. So for instance, this could be Vafidemstat or BHB or another therapy directly onto human cells to see the impact. We're so excited to share that we've already secured $20,000 toward this goal, and now we have $50,000 remaining to fund this critical step. As I said earlier, I'm not a scientist, but when I hear this, this is how I think about it. We put human skin cells into a petri dish, and we sprinkle, or maybe drown, those cells with the therapeutic, whether it's BHB, Vafidemstat, or something else. And we observe the impact of this introduction onto the human cells. The amazing thing is that by creating this set of cell lines, we can utilize this with a variety of different treatments. This isn't like a one and done, throw it out cost. We can establish these lines and use them again into the future. With an additional $50,000 through this effort, we will gather further data that can support our efforts to drive toward a clinical trial. As Charlene noted, this is one of those steps to prepare us for clinical trial readiness. Every donation helps us reach our goal, whether it's $10, $25, or more. We truly value every donation as it drives our therapy development, and we want to thank each of you for your continued involvement in KSF. I'm sharing this next slide, and feel free, here's the QR code. I think we've also shared the link in the chat to, if you want to contribute and support this work today. I'm also sharing here a variety of giving options on this next slide. If you want to make a donation in a different way, feel free to reach out to us if you want more details. Because of your past and continued generosity, hope is no longer just an idea. It's truly becoming a reality for us. And every gift you've made and will continue to make is fueling breakthroughs, lifting our families and bringing us closer to a day when lives look entirely different. On behalf of all those whose futures shine brighter because of each of you, including Brody, I want to thank you for standing with us and for making the impossible possible. And now I'm going to hand it back over to Pat.
Thank you, Makai, and thank you to everyone from the KSF team for pulling all of this information together that we shared here today. I hope you've all found it very valuable. And also many thanks to Charlene for joining us and sharing her journey and how crucial it is for patient advocacy groups to be driving research that will lead to therapies for rare diseases. As you saw from all of the work we shared today, KSF is active on many fronts. With your support, we are funding research grants globally to collaborate with leading researchers wherever they may be. Similarly, we are partnering with clinicians globally to decline the care guidelines that everyone with Kabuki syndrome will be using with their doctors, therapists, and other care providers. Through KSOC, we are laying the important and necessary groundwork for clinical trials of potential therapies. We are also working to grow the Kabuki syndrome community through initiatives like Kabuki Count and the new ICD-10 code, which will help us identify as many individuals with Kabuki syndrome as possible so that we have greater influence with pharmaceutical companies, biotech companies, and other research organizations so they will focus some of their efforts on Kabuki syndrome. I could go on. But the final message I want to leave you with today is that we cannot do this without all of you. Your support of KSF is critical, whether that's through participation in KSOC, completing surveys that guide and inform our work, helping to identify others with Kabuki syndrome, and we know there are many more to be identified, or funding us through your donations. We need your support. Likewise, please let us know how we can support you in your Kabuki syndrome journey. To that end, there's a link to a very short survey in the chat, and we'd really appreciate your feedback on today's event. Your input helps us plan meaningful events for you, and we certainly want to be doing many more of these in the future. So to wrap it, thank you very much for joining us today, and we look forward to sharing more updates with you all in the near future. Have a great day.